Malaria is an insect-borne infection disease that seriously threatens human health and is listed as one of the 3 major infection diseases for prevention and control by the World Health Organization. It has distinct clinical characteristics. Patients usually experience periodic chills, fever, and sweating. The fever reduction process, like the body's hot and cold switch, is controlled by the disorder. In severe cases, it can cause anemia, splenomegaly, and even coma and death. In history, malaria has been raging for a long time, bringing heavy disaster to mankind. Today, many people around the world are still suffering from malaria every year, and the prevention and control situation is still grim. Anopheles mosquitoes play a key role in the transmission of Plasmodium parasites that cause malaria. When a female Anopheles mosquito bites a patient infected with the malaria parasite, the parasite enters the mosquito's body with the blood, where it undergoes sexual reproduction and after a complex series of developmental stages, forms an infection sporozote. When the mosquito bites a healthy person again, the sporozote is injected with the mosquito saliva, begins to develop and multiply in the liver, and then invades red blood cells, causing them to rupture, releasing more malaria parasite and metabolites, triggering a chain of immune responses that leads to malaria. Anopheles mosquitoes, as the only intermediate host of Plasmodium, are widely distributed in tropical and subtropical regions. They are nocturnal and live in dark and humid environments. The ability of different species of Anopheles to transmit malaria varies. For example, Anopheles gambio is the primary factors of malaria transmission in Africa. Plasmodium is a class of single-celled eukaryotes, and 4 species of Plasmodium vivax, Plasmodium falciparum, Plasmodium malaria, and Plasmodium ovale are known to infect humans. Malaria caused by Plasmodium falciparum is the most dangerous, rapidly destroying large numbers of red blood cells and causing serious complications. The synergistic interaction between Anopheles mosquitoes and Plasmodium poses a great challenge to malaria control. After people learn about malaria infection, they will also want to know the disease that is caused by malaria. Black water fever and cerebral malaria are 2 serious diseases closely related to malaria. Plasmodium falciparum proliferates in red blood cells, causing red blood cells to rupture on a scale far beyond the normal metabolic load of the human body, and a large amount of hemoglobin is released into the blood. In the process of metabolism of hemoglobin, the accumulation of metabolites exceeds the excretion capacity of the kidney, which will cause the color of urine to turn black and lead to acute renal failure and the formation of black water fever, which seriously threatens the life of patients. In the pathogenesis of cerebral malaria, when the malaria parasite infects red blood cells, special proteins appear on the surface of the infected red blood cells. These proteins make the red blood cells easily adhere to the cerebral vascular endothelial cells, causing cerebral blood vessel blockage and preventing blood circulation in the brain. The brain due to ischemia and hypoxia, causing brain dysfunction, patient will have severe headaches, convulsions, coma, and other symptoms. If not timely treatment, the death rate is extremely high. The annual mortality rate of malaria remains a serious problem in the field of global public health. According to the World Health Organization, even with the continuous improvement of prevention and control measures, malaria still kills hundreds of thousands of people every year, especially in Africa, where children under the age of 5 are the main victims. Fluctuations in malaria mortality are closely related to the allocation of control resources, malaria resistance, and differences in healthcare. Due to the lack of effective prevention and control measures in some remote areas and the lack of self-protection awareness among residents, the risk of death from malaria remains high, and the prevention and control work remains a long way off. Next, the reader will learn about Plasmodium's life cycle in the mosquito. According to diagram 1, people can see that when a female Anopheles mosquito bites a malaria-infected person, the microgamete and microgamete in the infected person's blood flow into the Anopheles mosquito. In the mosquito main gut of Anopheles mosquitoes, small gametes swim quickly and combine with large gametes to form zygote. The zygote is transformed into an oocyte by development. By their motor ability, the zygote penetrates the midgut wall and develops into an oocyst on the intestinal wall. During the development of the oocyst, the internal cells divide and differentiate continuously. When the egg sac is mature and bursting cyst, it will release large sporozoites. The sporozoites circulated through the blood of the Anopheles mosquito and enters the salivary glands. When Anopheles mosquitoes bite a healthy person again, the sporozoites enter the blood with saliva, starting a new cycle of infection. Additionally, people will be taken to learn the plasmodium in the human body by also watching diagram 1. When an Anopheles mosquito carrying the sporozoites bites a healthy person, the sporozoite is injected into the blood with saliva, quickly circulates to the liver, and invades hepatic cells. The sporozoites of some plasmodium species are converted to hypnozoites, which is the reason for the recurrence of Plasmodium vivax and Plasmodium ovale. The rest of the sporozoites develop in hepatocysts and gradually form mature liver schizoids. Mature liver schizoids contain many merozoites. When the merozoites matures, the liver cells rupture and the merozoites are released into the blood. The merozoites that enter the blood then invade the urocyte and develop into trophozoites inside the red blood cells. The trophoblasts continuously absorb nutrients from the red blood cells and grows and develops into the urocyte schizoids. When the merozoites in the red blood cells mature, they rupture the urocyte, and the released merozoites continue to infect all the red blood cells and multiply again and again. In this process, the merozoites no longer proliferate, but develop into urocyte and gametocyte, preparing for the introduction of malaria parasite into Anopheles mosquitoes and opening a new cycle of transmission. Next, people will discover the Anopheles life cycle in detail, and we will also discuss the point in the life cycle where the species is most vulnerable. The life journey of the Anopheles mosquito, commonly known as the dark mosquito, consists of 4 stages: eggs, larva, pupa, and adult mosquito, as shown in the diagram 2. And the whole process is full of complex changes and adaptation strategies. When Anopheles mosquito lay their eggs, they prefer clean, slow-moving bodies of water, such as rice paddies, ponds, or the edges of streams. The eggs produced have a unique shape, a boat shape, and a floating sac on the surface, which allows the egg to float steadily on the water. These eggs enter the larval stage after 2 to 3 days of incubation, under suitable temperature and humidity conditions. Anopheles larvae have a relatively large head, a pair of distinct antennae, and a slender abdomen. In the larval stage, they get oxygen from the surface of the water through breathing tubes and feed on algae, bacteria, and organic debris in the water. During this period, larvae grow rapidly and need to undergo 4 molts, and their size will increase significantly after each molt. This phase usually lasts 7 to 14 days, depending on environmental factors such as water temperature and food availability. The larvae enter the pupa stage after the final molt. The comma-shaped pupa do not feed but can swim through the water with the help of a pair of breathing horns. This is a transitional stage in the development of Anopheles mosquitoes, usually lasting 2 to 3 days, and the pupa shell protects the internal tissues from complex reorganization in preparation for the emergence of the adult. When the pupa reaches maturity, the shell breaks and the adult emerges from it. Newly feathered Anopheles mosquitoes have soft wings that take time to harden before they can fly. Female Anopheles mosquitoes bite humans or other animals for protein to promote ovary development and transmit the malaria parasite, where male Anopheles mosquitoes feed on nectar and plant sap. In the entire life cycle of Anopheles mosquitoes, the larval stage is the most vulnerable. Currently, they live in a relatively fixed water environment, and it is difficult to actively avoid external threats. The use of insecticide or biological control measures, such as the release of mosquito fish and the introduction of Bacillus thuringiensis, can effectively eliminate many larvae. In addition, environmental modification measures such as chlorine water and filling photoliths to destroy the living environment of larvae can also control the breeding number of Anopheles mosquitoes from the source and effectively reduce the risk of malaria transmission. After people understand the causes and negative effects of malaria, they also need to know the prevention and treatment of this disease. To elaborate, the prevention and control of malaria requires multidimensional and comprehensive measures. The use of insecticide-threatened mosquito nets can form a physical barrier during sleep, prevent Anopheles mosquitoes from contacting the human body, reduce the risk of bite, and causes a relatively low, simple operation suitable for large-scale promotion. Indoor residue spraying is also a common means. Spraying insecticide on walls, ceilings, and other resting surfaces of Anopheles mosquitoes can directly kill the Anopheles mosquito in contact with the agent, and the effect is lasting. However, long-term use of insecticide may lead to resistance in Anopheles mosquitoes and cause some pollution to the environment. In addition, environmental control measures such as reducing water accumulation and cleaning waste containers can destroy the breeding sites of Anopheles mosquitoes and curb the breeding of Anopheles mosquito from the source, which is environmental friendly and long-term resources by the community. Additionally, artemisinin-based combination drugs are the main treatment for non-severe malaria, which has a fast effect, high cure rate, and relatively small side effects. However, in some areas, the malaria parasite has developed resistance to it, affecting the effectiveness of treatment. In addition, for travelers to malaria endemic areas or people in high-risk environments, it is often necessary to take anti-malaria drugs such as chloroquine, mefloquine, antifolins, and doxycycline. For example, chloroquine interferes with the metabolic process of Plasmodium and inhibits its growth and reproduction, thus achieving the anti-malaria effect. In addition, antifolin drugs are a class of drugs that work by inhibiting the action of folate in the body. They are often used in malaria treatment because they interfere with the growth and reproduction of the malaria parasites. Lastly, with the severe spread of malaria, the government should also take the initiative to create projects that can reduce levels of malaria. For example, the government can lead the application of transgenetic malaria prevention and control and Anopheles mosquito sterilization projects with the help of gene technology and insect sterilization technology to curb malaria transmission at the root and protect people's health. During the implementation of the project team will deeply cooperate with scientific research institutions to breed genetically modified Anopheles mosquitoes carrying specific genes. On the one hand, these genes can reduce the susceptibility of Anopheles mosquitoes to malaria parasites, blocking malaria transmission, and on the other hand, enhance their reproductive ability. When the genetically modified Anopheles mosquitoes are released into malaria endemic areas, they mate with wild Anopheles mosquitoes, allowing anti-malaria genes to spread widely throughout the population. At the same time, the captured male Anopheles mosquito was sterilized by radiation or chemical drugs, and then the sterilized male mosquitoes were released into the natural environment to mate with wild female Anopheles mosquitoes, reducing the breeding number of Anopheles mosquitoes and curbing the transmission of malaria. This project has significant advantages. It can effectively control malaria. Genetically modified Anopheles mosquitoes change the genetic structure of the population, reduce the susceptibility to the malaria parasite, and sterilizing Anopheles mosquito directly reduce the Anopheles mosquito population, protect public health, and reduce the medical burden. Moreover, compared with traditional prevention and control methods, which require a continuous investment of resources, this project is expected to achieve long-term malaria control by changing the genes and numbers of Anopheles mosquitoes and fundamentally reducing the obstructions of malaria to social and economic development. However, there are potential risks that occur as well. The release of genetically modified and sterilized Anopheles mosquitoes may upset the ecological balance. Anopheles mosquitoes have a specific role in the ecosystem, and significant population reduction or genetic changes will affect the organisms that feed on Anopheles mosquitoes, thus affecting the stability of the entire food chain and ecosystem. In addition, the continued use of specific genes or sterilization techniques may produce adaptive changes in the Anopheles mosquito population, reducing the effectiveness of the project. For example, Anopheles mosquitoes may develop resistance to sterilization treatment, resulting in decreased sterilization efficiency, and wild Anopheles mosquitoes may also produce new populations with stronger survival ability after mating with genetically modified Anopheles mosquitoes.